Let's dive right in. Today, we're unpacking a genuine medical mystery, one that's literally flipping our understanding of human metabolism upside down. For years, this supposedly miracle metabolic hormone, FG21, has been celebrated as an absolute hero for fighting obesity and metabolic syndrome. But recent groundbreaking research has uncovered a pretty dark side. It turns out, under stress, this exact same hormone might secretly be driving heart failure. It's a wild paradox, and we're going to decode exactly how this happens right now. Here's our roadmap for this explainer. We'll start with the FGF21 Paradox, move into liver and heart crosstalk, look at how prolonged signaling drives toxicity, examine the loss of the oxytocin shield, and finally wrap up with halting the hypertrophy. Okay, Section 1, the FGF21 Paradox. Let's look at the dual nature of FGF21. So here is the fundamental question driving this whole conceptual shift. Can a so-called miracle metabolic hormone actually break a heart? You know, in pharmacology, we absolutely love a good protagonist, right? And fibroblast growth factor 21, or FGF21, has long been exactly that. It's a master regulator of systemic energy homeostasis, and its analogs are even being used in clinical trials right now to treat obesity and lower hepatic fat. But what happens when the heart is put under severe, prolonged mechanical stress, like with high blood pressure? Well, this new research shows that our heroic protagonist slowly, insidiously, turns into the villain.
It turns out FGF21 kind of has a split personality. In the short term, you get these well-documented metabolic benefits, like normalizing circulating glucose, lowering body weight, and combating obesity. But over time, a highly controversial dark side emerges. It seems prolonged F21 signaling actually drives left ventricular hypertrophy, which is a dangerous thickening of the heart walls, and it's directly linked to heart failure. We're looking at a hormone that, under the wrong conditions, literally stops protecting the body and starts swelling the heart muscle.
Now you might be wondering, is this just some weird anomaly found in lab mice? Absolutely not. When researchers looked at human patients with systemic hypertension, folks who hadn't even developed full systolic dysfunction yet, they found that their cardiac FGF21 mRNA levels were a staggering 15-fold higher than in healthy individuals. 15-fold. That is huge, and it proves this is a very real, very potent human phenomenon.
Moving on to Section 2, liver and heart crosstalk. Let's trace where this toxic flood is coming from.
What we're looking at here reveals an incredible biological game of telephone. When the heart experiences pressure overload, say from uncontrolled high blood pressure, it causes a backup, basically a hemodynamic congestion in the liver. The liver senses this mechanical stress, panics, and hits the big red button, dumping FGF21 into the bloodstream as an SOS signal. But when that massive wave of liver-derived FGF21 finally hits the heart, it does something entirely unexpected.
It actually triggers a biological chain reaction known as hepatocardiac crosstalk. The endocrine signal from the liver forces the cardiomyocytes, the actual heart muscle cells themselves, to start producing their own localized FGF21. So it shifts from being a systemic alarm to a really dangerous, localized autocrine loop. The heart is essentially being told to swell up and remodel itself, driven by the very hormone it was just tricked into producing.
All right, Section 3, prolonged signaling drives toxicity, because time is the critical factor here. Let's trace how this toxicity actually builds across a very specific biological timeline. Just 3 days after cardiac pressure overload begins, the liver starts releasing its FGF21 into the plasma. At this point, the heart's own FGF21 is totally normal. But at the 2-week mark, we cross a critical threshold. The heart's FGF21 production spikes, and we see the onset of left ventricular dysfunction. Finally, by 8 weeks, pathological hypertrophy has fully peaked, bringing massive fibrosis and inflammation right along with it. And at that 8-week mark, the heart doesn't just look different on the outside, its internal engine has entirely stalled. We witness a severe metabolic crash. There's severe cardiac glucose depletion, meaning the heart has lost its primary clean-burning fuel source. Instead, we see this elevation of branched-chain amino acids, which is really just a desperate attempt to find alternative energy. Because of this, the production of ATP and NAD+ completely plummets. It even depletes cardiolipin, a crucial structural lipid inside the mitochondria. The heart's engine is essentially running out of the right fuel and breaking down on a microscopic level.
Honestly, this is the ultimate biological definition of too much of a good thing. While a short, transient spike of FGF21 might be an adaptive attempt to help the body deal with stress, prolonged FGF21 exposure forces a devastating cellular reprogramming.
It's the prolonged signaling itself that drives the toxic left ventricular hypertrophy and pushes the heart straight toward failure.
This brings us to Section 4, losing the oxytocin shield. Let's look at the hidden victim in all of this. Now, this maps out the interference perfectly. Normally, the brain's hypothalamus produces a neuropeptide called oxytocin. And yes, that is the exact same hormone famous for social bonding and childbirth. Well, in the heart, oxytocin acts as a powerful cardioprotective shield. It naturally prevents heart muscle cells from swelling uncontrollably. But, as this research shows, prolonged high levels of systemic FGF21 actually suppress the hypothalamus, meaning it literally cuts off the brain's supply of protective oxytocin to the heart.
The receptor levels really tell the whole story here. In a normal control heart, oxytocin receptors act at 100% capacity. But put a normal, wild-type subject under extreme pressure overload, and those receptors crash down to just 45% because of that FGF21 flood. But here is the incredible part. In subjects where researchers genetically knocked out the liver's ability to produce FGF21, the heart's oxytocin receptors stayed beautifully preserved at 95%. By silencing the liver's false alarm, they actually saved the heart's natural shield.
To prove this wasn't just some random coincidence, the researchers took heart cells and bathed them directly in FGF21. As expected, they swelled right up. But when they added oxytocin to the mix, the cells stayed perfectly normal. The researchers definitively concluded that oxytocin attenuates the pro-hypertrophic effect of FGF21. Basically, oxytocin directly fights back against the physical swelling, proving that FGF21 destroys the heart in large part by simply disarming its best defense.
And finally, Section 5, halting the hypertrophy. Let's talk about a real-world cure.
So, all this medical mystery actually translates into a tangible, real-world cure. Using a targeted therapy called antisense oligonucleotides, or ASOs for short, researchers successfully sought out and silenced the FGF21 mRNA. And the tissue samples prove it works. The heavy, damaging fibrosis typically seen in a failing heart is massively reduced after treatment. The heart tissue is essentially rescued.
Let's walk through the exact 3 steps of how this intervention cures the pressure-overloaded heart. Step 1. Systemic inhibition of FGF21, using those ASOs, which quiets that false alarm coming from the liver. Step 2. Because the FGF21 flood stops, the hypothalamus wakes back up, leading to the full restoration of the heart's oxytocin shield. And finally, Step 3. Without the autocrine FGF21 driving growth, and with oxytocin fully back on guard, we see a dramatic reversal of cardiac hypertrophy, inflammation, and fibrosis.
The implications of this are absolutely massive. We're talking about a targeted ASO therapy that completely stops the destructive hormonal chain reaction. This offers a true paradigm shift for treating pressure overload heart failure. For decades, we focused almost entirely on treating the mechanical issues of high blood pressure, but now we have a blueprint for stopping the underlying metabolic crosstalk that actually causes the heart to fail in the first place.
Which leaves us with this final, really provocative thought. We spend so much time trying to boost our metabolism and enhance hormonal signaling for better health. But as we've seen today with FGF21, could turning off our body's own overactive metabolic alarms actually be the hidden key to saving our hearts? It's a fascinating look at the delicate balance of human biology, and a stark reminder that in medicine, the line between a miracle cure and a hidden danger is sometimes just a matter of time. Thank you so much for joining me on this explainer, and I hope this completely changes how you look at the complex systems beating inside your chest.